Put it on my phone. On my computer. I'm on my charger up for real.
Alrighty, so I'm going to be turned around here a little bit so you guys can see. So we'll see how this works. All right. I'm glad I've always put everything. Okay. So when we left off, we were talking about glucose. So like I say, I've got the exam. Everything looks good so far, as in terms of there's stuff there for me to grade. Everybody's on good terms. I'm happy. I'm pleased. People are taking the exam. It's much better than my freshman class where it's like, yeah, I'm just not going to take the exam and not tell anybody about it. So, but yeah, so everything's good. I'll work on making sure everything's clear for the next exam, which won't be that far ahead, because the good news is that was the worst exam. That's the toughest one by far. This one coming up shouldn't be too bad. We've already talked about glucose and the structures in there. We talked about disaccharides and polysaccharides, and we can see things like agarose here. I think this is where we ended up, where we have this nice little heteropolymer. So maybe, there we go.
Not fun.
Sorry?
I can't find the PowerPoint.
It's under module 3, yes. The first PowerPoint.
So our polymers, we can get more and more complex, okay? So we can have a variety of things. I always kind of joke, what's the difference between a proteoglycan and a glycoprotein? It's kind of like which came first, the chicken or the egg. So we're going to talk in kind of general terms about these today. Now, instead of having like say amylose, where you just have a repeating monomer of glucose, we now take a disaccharide and repeat that over and over again. Okay. For our glycosaminoglycans, one of these is either N-acetylglucosamine or N-acetylgalactosamine. Okay. They tend to be negatively charged. We'll get our uronic acids and our sulfate esters and all the good stuff in there. They can be very hydrated to help minimize charge repulsion in there. If all those negative charges together, they're going to push each other apart. So we put a lot of water in there to hydrogen bond with that. So again, keep in mind, hydrogen bonds answer to everything. We'll see our glycosaminoglycans form a kind of the basis of our extracellular matrix. If you think of one of the things that the extracellular matrix does is it provides structure. It also provides cushioning. We're going to talk a little bit about your knee joint because this hydration of one of the particular aminoglycans in the knee joint, is it hyaluronic acid? We'll see it here in a minute, allows you to have this flexion there. It kind of functions like a gel, like those gel inserts into your shoes in there. So we'll see a lot of these, carbohydrate attached to proteins that'll function in connective tissue or joints. Okay, so if we look at just kind of pictures of these, this would be our acetylglucose, glucosamine, and N-acetyl glucosamine. This stuff we have up there. So this is our hyaluronic acid. Our molar mass is going to be huge. We're going to have about 50,000 disaccharides per chain. If you look at chondroitin sulfate, we're only talking about 20 to 40 disaccharides in that. So it tends to be much smaller. If you ever go to, like, was it GNC or any of those supplement shops, and take chondroitin sulfate for your joints and whatnot. And I don't know how effective that is, taking it that way, but you certainly need these in your extracellular matrix for flexion and movement and stability. We also have some others in here like keratin sulfate and heparin. We'll see heparin not only in ECM in terms of structure and shock absorption, but also in terms of controlling bleeding as well too. All right, so coagulation and blood clotting. Just to visualize a three-dimensional segment of it, this is kind of what it looks like. So you have this nice little springy section in there. Heparin and heparin sulfate. Our heparin is actually a small polymer, about 3 to 40 kilodaltons. Heparin sulfate is when you take this polysaccharide and attach it to the proteins. Okay? It has very high negative charge density, and it works to prevent blood clotting. it activates this antithrombin, which prevents thrombin from stimulating blood clotting activity. The other neat thing about heparin sulfate is it can bind to cells. If you look at, say, like, for example, I have a little road rash here. I learned to do not play basketball with my wife because she is mean and cheats. You know, she sent me to the urgent care on Monday by tripping me. I have bruised ribs, fortunately not broken ribs, but bruised ribs, because for the first time since I was a kid, I fell and did not land properly. Need to take more Kimi and Aikido. That's what I've been missing out there too. But when these blood vessels regrow, you know, our heparin sulfate is going to help regulate those. Okay. The other thing we can see is a lot of our, you know, our glycosylated proteins can bind to various invaders in the system, like viruses and bacteria, and help decrease their virulence by making them less able to fuse with the cell membrane and invade. Okay, if we look at just kind of like a three-dimensional picture here, we have our thrombin and our antithrombin kind of together with our heparin kind of regulating the activity in there. All right, we have this lovely table in here, which is way more information than you need to know. But if you look at like the first three, yes, the first three, you should probably have a good idea about our amylose, amylopectin, our glycogen, and our cellulose. Those are the ones that we talk about most. They're all glucose oriented. It's just whether they are branched or unbranched, how frequently those branches are, and whether they're in an alpha 1,4 connection or a beta 1,4 connection.
So we only need to focus on the first three?
Yes, yeah. I guess you could count it 4 because amylose and amylopectin. I might ask, what's the difference between amylose and amylopectin, or amylose and glycogen, or glycogen and cellulose, or any one of those combinations in there I could come up with. All right. And also, I'm going to preach one. Yeah, actually, let me go ahead and preach now too. So I know we've mentioned cellulose. Seems like, why can't I go out there and chew on some leaves or make some grass soup or something like that and digest it? It's because we lack the enzymes to digest cellulose. There are some bacteria that are in a symbiotic relationship with, say, like ruminants and termites that have that cellulase enzymatic activity that can digest it. So that's why you'll see, you know, cows chewing their cud so that they can expose it to this bacteria and help break it down. So a few years back, I saw a video that had been smuggled out of North Korea. Basically, you know, there's some people there, and there's this girl, probably about your guys' ages, who was walking along the road, and she was kind of disheveled and a bit confused and something like that. And so they kind of filmed this interview with her. She was just wandering around kind of confusedly. Turns out, you know, so North Korea, a lot of famine, a lot of not much access for food, especially if you are from a rural area and not part of the ruling party. So her parents had apparently died, and she was wandering around begging for food. And there just wasn't a community that could help her. Nobody had food, so she was wandering around there. What a lot of the poor people, as they refer to them as the peasants, because apparently we still have peasants in this day and age, would do when they were hungry is they would make grass soup. So they, you know, just basically boil some grass and kind of chew it up. It has no nutritional value, but it makes your stomach feel full. But if you have a diet of this, you know, for a month or so, and that's all you have, then eventually you're going to starve to death and die. So I forget who it actually was in here, but this is kind of one of these things where they weren't supposed to be talking to people like this, but they were. And, you know, it was very touching because she seemed like a very normal student, but she was just confused. So yeah, it's a compelling video. I'll have to find it and post it. Yeah. But the same journalists who managed to record that, you know, were there a month or two later and, you know, searched for her, and she had died of starvation, which just seems crazy. Even in the food deserts that we have here and the lack of food security we have, even here in Winston-Salem, it's very rare that you will see someone who actually starved to death. And this was something that was actually fairly common during the famines in North Korea, because there is, you know, outside the party enclaves, there's no healthcare concern in there. So, you know, what could we do, you know, to allow us to eat grass? Maybe we could come up with a way to, you know, transfect cellulase into human beings, kind of like my idea of transfecting photosynthetic genes into humans as well too. that's getting a little too crazy into out there. But you know, it's something to think about in terms of starvation and food security and how we can actually increase access to food. Anyway, so our structures become important. If we look at some other glycoconjugates that we have out there, there's a whole class. Basically, you can think of our carbohydrates in a way as like decorations. You know, you see all those little stickers up. This type of glycoconjugate is going to be our glycoproteins. So a glycoprotein is a protein that has some small oligosaccharides attached in there. Now I see that my internet connection is telling me it's unstable. Why? I tell you, yeah, put all this crap on your computer and make everything slow, and then we still won't let you connect. All right. Anyway, glycoproteins, we have a protein with small amounts of carbohydrates on there, as opposed to a proteoglycan, which is going to be a carbohydrate with small amounts of amino acids attached. So our carbohydrate usually gets attached with its anomeric carbon, and it will be attached to the amino acid on there. This most commonly happens at serine, threonine, or asparagine. This is where, and I won't put you guys who are actually here on the spot, but I'd say, why do they connect to serine, threonine, or asparagine? And this is where you have to think about, okay, what do they have in common? All right, let's divvy that up. What do serine and threonine have in common? You guys remember the structures for those? Their side chain has a hydroxyl group, right?
Oh, yeah.
Yep. So we can have a linkage to that oxygen. So our anomeric carbon can attach that oxygen. What about asparagine? Well, it's got an amine on its side chain. So we can have either O-linked or N-linked chains here. And like I say, we'll see more on that in greater detail later on down the road.
Oh, I'm sorry. So you said serine
and threonine have that hydroxyl group side chain. So our carbohydrates will attach to that oxygen. Basically, that oxygen, that hydroxyl group will donate its proton, and you'll get the hydroxyl group from, say, glucose, and that's where your condensation reaction occurs to give off water, and you have this O-link in there. You can do something similar with the amino group of asparagine, where the amino group will donate a proton, and the hydroxyl group gives off from say glucose gives off the OH group. You get another condensation, but you have this linkage now to a nitrogen instead of that. So that's our O-linked and N-linked. At least we name them, you know, fairly straightforward. This is normally something that happens primarily in mammals. Roughly half of our proteins are glycosylated. So it's kind of like you think of like running into, you know, kids in high school. It's like, how many girls wear makeup in middle school or something like that? And so you get the point where it's like, well, half, you know. So half of our proteins are glycosylated. We add this enhancement in there. And there's a whole variety of roles our glycoproteins can play. One of the neat things is very few bacteria can glycosylate proteins. So they have their kind of carbohydrate cell walls and their protein separate in there. Our carbohydrates will often play a big role in protein recognition. We can see this in our blood types. Type A, B, AB, and O are based on the presence or absence of a pair of carbohydrate groups in there. Turns out viral proteins are very heavily glycosylated, and that helps them sneak past the immune system. It's like, they look like they belong. They're carrying a clipboard, so no one questions them. That's an old military joke. All right. So here we see our serine threonine with our O-linked attachment, and here we see in this with our N linkage on there. And again, you know, we have some lovely color coding on there to indicate which sugars are partaking in this, but you don't need to know all that good stuff in there. Notice it kind of gets pretty crazy pretty fast. It's like, what's this purple triangle? You know, purple diamonds, you know, green circles, blue squares. It's like Lucky Charms, right? And it's like, yeah, they're after me carbohydrates. Terrible. All right. We have another class of glycoconjugates. These are our glycolipids, all right? And these are lipids, which we're going to talk about next, that have bound oligosaccharides. A lot of these are found in plant and animal cell membranes. We take our lipid components in there and we attach some sugars to them. Okay? So for vertebrates, the ganglioside composition determines our blood groups. Okay, so kind of what I was talking about before, but we'll take a peek at that. And in bacteria, we can have our lipopolysaccharides that will cover the peptidoglycan layer. So it's basically another blanket over top of that. Basically, the gram stain can't hit the peptidoglycan layer because that lipopolysaccharide protects it. All right, we can get some crazy stuff in here. Here we have this big lipid core, and here we can have our O linkage. Again, we can have N and O linked carbohydrates attached to our lipids. Notice our lipids are a lot crazier shape than our amino acids generally. Some examples of those coming up here very soon, today. Our third class of glycoconjugates that we kind of divide things up into are proteoglycans. It's like, wait, glycoprotein, proteoglycan, yeah, chicken and egg. Our proteoglycans are basically large chains of carbohydrates with peptides attached to them. We can further divide these into different subclasses, like our syndicans, our glypicans. We have a protein has a single transmembrane region. A glypican has an anchor to a lipid membrane. It's better to actually look at this pictorially. All right, so we see something that looks like this. Here's our syndican, which goes across the membrane. Here's our glypican, which is attached to a chain. Our GPI anchor in there was it? Something phosphoinositol. Yeah, maybe something, but it's basically attached in there. The neat thing about this is we see there's cleavage sites in here. We can have enzymes that could come in and cut their connection to the membrane region, and we can isolate them that way. You can see our syndican, we might have strands of heparin sulfate coming off of that. We might also have chondroitin sulfate. So we're going to see some particular patterns coming up here as well. There are these particular binding regions in here within. Basically, our heparin sulfate has little sections that kind of repeat over and over again. So you can add more stuff to that, and you can add more stuff to those links. Oops, wrong slide. But we'll see that. And it's basically kind of like one of those fractals where you draw a line and you draw a line coming off that, and you draw like 2 lines coming off that, and 2 lines coming off that. So you basically get this big expanding tree shape. All right. So our different proteoglycans are going to have different glycosaminoglycans attached to that core protein. Okay. All these linkages are going to deal with the anomeric carbon, of course. So knowing that anomeric carbon, you can see there's probably definitely going to be questions about the anomeric carbon and the attachment of carbohydrates to stuff. There's a lot of different core proteins, but aggrecan, named because it aggregates and forms big clusters, is probably the best studied. We can look at this terminus in here, but we're not going to sweat this too much. But again, we can see our different types of linkages, beta 1,3, beta 1,4, beta 1,3, beta 1,4, kind of repeating in there. But let's take a look at what these aggregates can do. We can get these huge aggregations of these proteoglycans that will hold a lot of water, so they can provide a lot of lubrication. They're very low friction. So this is the stuff that you're going to find in cartilage that will allow you to flex your knees, or if you say jump off of a step and land, that they function as kind of a shock absorber. If you ever put like those Dr. Scholl's gel inserts into your shoes and they're like, oh, it feels so bouncy and springy, that's essentially what's happening with these types of proteoglycan aggregates in there. If you look at an example of one here we have our core protein. We have these attached that are attached to the main rod in here. Then we'll see close to the main rod, we'll have our hyaluronic acid. Yeah, followed by our keratin sulfate coming off there in the blue, and then chondroitin sulfate as we get further away. We have these big kind of feathery sections in here, and you can see under the electron micrograph, it really does look like a big kind of feathery section in there. And that functions much like a little squeegeer or a sponge, because you can put water in there, and that water gets stuck in between all these little feathery parts in here, so it can absorb a lot of water and it's a big cushion basically
So
in
you
there.
said the proteoglycan aggregates are like cartilage, and they act as the shock absorber.
Yes, yeah. They basically reduce friction so that when you move, you flex your elbow or your knee, that you don't have like bone on bone or what's the word? A lot of stuff that's scraping together that's not good. If you think of like osteoarthritis, that's when the cartilage breaks down over time and you get scar tissue in there and you start to get joint pain. That's because you're lacking some of this ECM material that will allow that easy flexion. Oh, that makes sense? Good. All right. Awesome sauce. Okay, let's talk a little bit about the extracellular matrix, because that's something that we don't talk about a whole lot, and usually, that's like more, advanced grad school, maybe in your, maybe in your sophomore year, but typically in maybe your third year. If you're going into this area, we study more about it. And this is all the stuff that's outside the cell. We almost exclusively focus on the cell and all the processes that take place in the cell, especially in introductory biochem. But we should know a little bit about what's outside as well too, because the ECM gives us kind of our shape and our structure. It's our production, and I would say in a lot of ways it's analogous to the girders and the things that hold up a building. Say, like when the new science building was being built out there, probably would come down to campus and you'd see like the girders up there, or they're putting in all these connections and they're welding stuff. Our ECM is kind of that invisible structural component, and then the cell is where all the stuff happens. So we can walk into the classroom in there and turn on the lights or use some equipment or whatnot. But it's the girders in there that help give it its shape. It also functions to give strength, elasticity, and function as a physical barrier. So that's why a lot of times something that may infect your kidney can't infect your liver, because it has, there's this ECM barrier between the two. The main components of this are things like proteoglycan aggregates, collagen fibers. Collagen plays a huge amount in there.
So since you said ECM has a barrier that prevents, like, I guess you said a tumor from getting to other tissues. So how do diseases spread to other parts of the body?
So a lot of times you'll see they will travel through the bloodstream. So that's when we look at, and I want to draw back. I don't want to talk too much about cancer because I don't know as much oncology. Yeah. You get that whole metastasization travels through there, and and then it can infect another area that way. Same way with, I think, viruses and bacteria. But, and I'm leading up to something in here as well. Yeah, I think we talked about tumor cells here too. Yeah. So some tumor cells can actually worm their way through there. If you've ever had strep throat, group A streptococcus, you still have strep throat, because one of the things that group A strep can do is it can actually senesce, and it goes into the interstitial layer, basically in the ECM outside of the cells in there, and it just kind of sits there dormant. You always have a few group A strep kind of sitting around it. Opportunity in this case, they can come back into the cells, reinfect. So, you get that sore throat again because the cells are being reinfected with group A. But you can't completely wipe out group A strep, because there are always going to be some that are going to be unable to be reached because they're hanging out mostly dormant in the ECM. So the ECM is kind of like a wasteland, it's as far as travel. Stuff can't get in or out too easily, but it definitely can't travel through there very easily, because they're, unlike say in the cell where you have those actin fibers in the cytoskeleton in there that kind of lead everywhere, and you've got import and export. You can put stuff into the extracellular matrix, but the ECM is generally more mechanically oriented and less transport oriented, aside from the circulatory system. So, I think that's a reasonable way to say that, although I'm not super confident on that. All right. So how do cells interact with the extracellular matrix? And this is why I really like the extracellular matrix, is because we essentially have this signaling system from our active stuff that's happening in cells to our support system and structural stuff there. Kind of like, when you have the tap in one of the lab rooms in there, you turn it on, that's our interaction, but our water pipes are actually in the interior of the walls and stuff. At least we hope they stay there. So we have this interaction between when we turn on the tap and in the water that's actually flowing through there. Or same way that maybe a light switch would work. So we have these integral membrane proteins that connect the exterior of the cell, the ACM, to the interior. Okay? I'm most familiar with integrins. Integrins are really kind of neat and cool because they are dimers, and basically when there's an interaction, I can't bend the right way here, but we'll pretend the table is the cell membrane and underneath the table is outside the cell, and inside the cell something binds. These basically kind of open up or close, so there's this conformational change like this, and these are attached to the cytoskeleton on the inside of the cell, and they're attached to usually collagen or elastin on the extracellular matrix. So if there is a mechanical strain out there, that strain can get converted into a signal that can help regulate whatever the cell is going to do, maybe produce more energy or something like that. But by this linkage, we can also moderate cell growth, cell mobility, apoptosis, wound healing. So I guess my mechanical stress of sliding across the concrete says, oh no, make ab and heal. I don't know. Visually, what this kind of looks like, here we have our collagen fibers and our proteoglycans and all of our aggregates that we can see up there. These are our integrins. And then we have our interior section here where we're attached to the actin filament. Something binds. We see those kind of split apart. So much like we saw myosin with its power stroke or walking along the actin filaments, we can see our integrins putting the strain on the actin filaments, which is going to have other intercellular effects. Yeah, so I guess just know that there's a communication system and it's usually moderated by integrins. All right. So there's a lot of ways that our oligosaccharides can participate in cell life, cell metabolism. And this is just a very brief introduction. I'm not going to go into any detail about any of this, but we're going to talk about glycolipids up here. Those are coming up in the next topic that we're going to cover, next chapter that we're going to jump to. We can have our proteins with their oligosaccharide chains attached. You can see a virus might recognize a particular oligosaccharide chain, and that's how it knows to bind to this particular cell to try and infect it. Same thing can come from a bacterium. We can also have maybe a leukocyte that's going to come in there and maybe activate a macrophage or something like that. Various toxins can bind into some of these signals in here. And of course, we can have our enzymes, enzymatic activity. We can have endocytosis that can lead something in there, and stuff to happen, and other stuff like this. So really what we're talking about here is our oligosaccharide chains play a key role in recognition. So it's basically our communication. Do these two match up? Can we bind to this? So we could talk about specificity and all that good stuff. In fact, one of the ways that you look at maybe treating some type of virus or bacteria is to change the specificity that it has for its particular receptor, or block its receptor, or stuff like that. All right. Now we want to keep this slide in here. It's like, no, too much. This is where we get to Lehninger and say, okay, thanks, that's enough. No more. So just to summarize this chapter, really kind of a short chapter. We talked about some monosaccharides. So, like ribose and glucose, galactose, and mannose. Good to know those structures and good to know how they interact with each other. And our disaccharides, like fructose or sucrose, or we can attach those two together. And then some polysaccharides like amylose, amylopectin, glycogen, and cellulose. And then what we covered today were just some various glycoconjugates and what roles they play. We're not going to get into any detail about their structures or stuff like that, but what do they do? How do they help a living organism stay alive? Okay, questions about any of this? All right, let me pause a second here because, it'd probably be good. Probably good if I'd just keep talking forever. I better check the time. I know we got more time here, but all right. Let's jump ahead. So this is where, whoops, wrong one. This is where we jump ahead in the textbook. We're going to go from chapter 7 to chapter 10. So this next exam is only going to have 2 chapters on it, 7 and 10. 8 and 9 both deal with DNA and RNA. That's why I keep them together. And then 11 and 12 about signaling and get us into some stuff as we end the semester here. All right, let's find this one. That should be this one. Okay, let's do it this way. There we go. Now, if I share my screen. Yay. Okay. So we can talk about lipids now. And there's all sorts of fun stuff with lipids. Lipids are fun. Lipids are what make bacon taste good. It's like when my son was very little, first time I showed him taking pictures of when he first tasted bacon. He's like, oh, this is so good. And then he was like, yeah, can we butter the bacon? And I was like, bacon makes its own butter. You don't need to put butter on the bacon. So anyway, but lipids play a lot of roles in here. So we're going to learn what some of those roles are. We're going to talk about their structure and properties. We're going to distinguish between storage lipids, which are fairly simple, and membrane lipids, which aren't that bad, but you can start mixing and matching and stuff. And you can make them super complex. We're not. We're just going to go into the edge there. And then we'll talk about signaling lipids as well, too, which you can think of in terms of like our hormones, our cholesterol-based compounds. Okay. So if we talk about lipids, they're a really diverse class of stuff. We can have a single chain here.
For some reason, Wi-Fi is not working for me.
Yeah, I'm kind of worried.
Mine's working.
Okay, it's vague. Yeah, don't worry, I'm not gonna go too far into here. Like another. But yeah. So we can see our simple fatty acids, our triacylglycerol. We can look at sphingolipids. We can look at cholesterol. We can also have glycerophospholipids. Then we get into some weird stuff in here, like what is this, ubiquinone and other semiquinones and fun stuff in there. So our lipids, generally, we can kind of summarize them by saying they're organic, they have low solubility in water, and they tend to be very hydrophobic. So as opposed to our carbohydrates that we just talked about, where they can suck up lots of water, our lipids are going to, be the opposite of that. They're going to stay away from water, repel water, right? Like water off a duck's back because of the lipids in duck's feathers. Okay. So what do lipids do? Right? We can think of storage of energy. So we talked about carbohydrates and glycogen for medium-term energy storage in the liver. Our lipids are going to be our longer-term storage of energy. So our fat cells, our adipocytes are going to store lipids for energy for a long period of time. The good news is they give us lots of energy. If you go back to, like, say, nutrition, maybe in like one of your early biology classes, you learn like what proteins and carbohydrates give you like 4 joules per mole or something like that, 4 kilojoules, 4 calories per mole. Yeah, and fats give you 9. So they are more than twice as much energy in there. The other thing is, because they're hydrophobic, you want to have a lot of water and to help together. They'll pack together nice and compact, so you can store a huge amount of energy in a relatively small space. The only problem is that relatively small space keeps expanding as you get older. So, all right. So they're good for energy, and I don't know if you've seen, like, one of my friends is really bear contest. So somewhere in Alaska, they have a bunch of grizzly bears, and they're packing on the weight so they can go through hibernation. So they're kind of doing like the March Madness playoffs. They have fat bears that you can vote for. Like, who's the coolest fat bear? I don't know, but it's weird. Anywho, our lipids can also function as insulation as well too. You can see this a lot of say like whales and dolphins will have layers of fat or blubber to insulate them from the cold ocean waters in there. Lipids have low thermal conductivity and high heat capacity. They can also function as mechanical protection. So I was joking, of course, when I took my little spill on the concrete, landed right on the ribs. Couldn't land on the stomach, which would have cushioned it more. Our lipids are also water repellent, so it keeps things like birds' feathers from getting too wet, so that they don't get weighted down and can't fly. It also can prevent the loss of water from evaporation. One we don't talk about too much, but buoyancy control and acoustics in marine mammals. So our whales, they can float by their lipid compositions in there. All right. We're probably going to talk a little bit more about membrane structure because we got our glycerophospholipids that we'll see in there, and our sphingosines, cofactors for enzymes, various vitamins. If you think of the vitamins that, you can't overdose on vitamin C because you'll just pee it all out, right? So all those vitamin C's to fight off the cold, maybe not so much. But you can overdose on which vitamins? Anybody remember? A, D, E, K, ADEC. So vitamins A, D, E, and K are lipid-derived vitamins. So they'll actually accumulate in the adipocytes, and you can overdose on those. Theoretically, you can overdose on vitamin C, and if you just guzzle it down there, you'll end up causing some type of damage or something. But it'll be hard. It's kind of like getting drunk off water. It's theoretically possible to drink so much water that you become intoxicated, but at that point, you're actually close to drowning just from drinking that much water. All right, but vitamin K, Q. We also have a lot of signaling molecules out here. We're going to have our paracrine hormones, which are going to interact, or going to induce changes usually within the same cell or within the same organ, and our steroid hormones, which can get into the bloodstream and have downstream effects all over the body. Various growth factors, vitamins A and D again there. Pigments. If you ever wondered why is a cardinal red and a canary yellow, it's a lot of their pigments are lipid derived in there. And actually, there's a kind of a neat thing with the cardinals. It's based on their diet. So the particular seeds that they eat help give them their brilliant colors. Right. And antioxidants. Again, I remember when I was a little kid and I had a bike accident, and the neighbors at the end of the road were very kind and gave me a Band-Aid, and they put vitamin E on my cuts. I was like, what is this weird stuff? That's an antioxidant to prevent infection. There we go. Our canthaxanthin for our bright red birds, and our zeaxanthin for our bright yellow birds. All right. So we're going to start by classifying some of these lipids because there are many types of lipids in here. We're going to look at complex lipids, ones that have fatty acids in them, and we can further separate those down into storage lipids and membrane lipids. And then we're going to talk about lipids that don't have fatty acids, like our cholesterol, our vitamins, our pigments, those kind of more unusual ones. So today we'll only get into the fatty acid ones, and we won't get too far through that. But if we kind of look at these, we have our storage lipids, which are generally going to be triacylglycerols. If you ever go into the doctor and they check your TAD count and your LDL and your HDL, all that good stuff in there, our triacylglycerols are basically a 3-molecule glycerol backbone with 3 fatty acids attached to it, and that's how your fatty acids are stored in the adipocytes as a triacylglycerol. And then those can be shuffled around through the bloodstream by various other components. When we talk about membrane lipids, we're mainly going to talk about phospholipids, okay? And we can have glycerophospholipids, which is, I'll often just say phospholipids and mean that. But our Our glycerophospholipids are going to have glycerol, just like our triacylglycerides, and they'll have 2 fatty acids, but then they'll have, in the third position, instead of another fatty acid, they'll have a phosphate head group, and then something attached to that. Could just be, another OH. It could be something more and more complex.
It can be any R group.
Yes, any R group. Yeah. Our sphingolipids physically look very similar to glycerophospholipids, but they have sphingosine instead of glycerol, and sphingosine has its own head, its own fatty acid tail group. It's going to have a second fatty acid attached by a nitrogen linkage, but then it'll have another phosphate group up there and something attached, maybe choline, maybe something like that. Glycolipids, we can stick sugars up here instead of our phosphates. Okay, and again, we can have glyceroglycolipids or sphingoglycolipids. When we get into archaebacteria, we can get into some weird stuff. There is a particular archaea organism that essentially makes rocket fuel. It's incredibly toxic. So they don't want it leaking out of the cells, and they're also in, I think, high temperature environments. So instead of having a phospholipid bilayer, they have a monolayer, but they have this glycerol on both sides of the monolayer. So it's much, I think it's called laterine. So it's a much tighter cell membrane so that stuff doesn't leak in or out. Yeah, but they make rocket fuel. All right. I think we'll see that later on. Let's talk about fatty acids, okay? So our carboxylic acids, again, we're having, our head group, our end is going to be a carboxylic acid, our COO, or COOH if we protonate it. Typically, these are going to be between 4 and 36 carbons. I don't think you'll ever see one less than 4 or more than 36. We probably won't talk about any more than 24. Almost all fatty acids have an even number of carbons, and most fatty acids are unbranched. So I don't think we're going to talk about any branched ones in here. So we can define our fatty acids either saturated or unsaturated. If they're saturated, you basically jammed on as many protons as you can. So there are no double bonds in the chain. The only double bond is in that carbonyl carbon to an oxygen. Everything else is like CHH, at the end. Monounsaturated has one double bond in there somewhere, and polyunsaturated has multiple double bonds. Oh, God bless my internet connection. What are you doing? What are you doing, IT? Is there really such an issue here? All right. But we'll see some better pictures of this as well too. So this is down here is a saturated fatty acid, and this would be a monounsaturated fatty acid. And this is actually going to be in the cis configuration as well too. We'll talk about cis and trans. If you ever wondered about trans food, yeah, we're going to learn about those too. Other fun stuff, nomenclature. We can be really efficient in naming these. I can remember struggling through early biochem going, what's the difference between oleic acid and olenic acid? And because there are all these names that derive from where the fats were first discovered and all that stuff in there. So we can have a systematic name. We can call something that's shown down here that has 18 carbons. So we start with the carbonyl carbon as 1, then 2, 3, 4, 5. You note that this is our alpha carbon, and number 18 would be our omega carbon. We could call this cis 9 because we have a cis double bond between 9 and 10. Cis 9 octodecenoic acid. And this is decenoic because it has that double bond in there. If it were a saturated fatty acid, this would just be octodecanoic acid. All right.
Do we have to do naming on tests?
So no, you want to do a name like that. However, we have this super efficient thing down here, right? 18.1 delta 9. I love this numbering system because it is a very concise way of telling you all the information you need. It basically tells us, hey, we've got 18 carbons. There's one double bond. That double bond starts at position 9. This little delta 9 up here tells us it's right here at 9. Okay. We could go back. So there's an omega numbering. If you've ever seen like, oh, take fish oil capsules. They have lots of omega fatty acids that are good for your heart. I don't know if you've seen those like that, yeah. Omega, we just number them from the tail end going this way. So it's the opposite direction, which we then 1, 2, 3, 4, 5, 6, 7, 8, 9. Turns out this would be 18,1 omega 9. Same thing here. I'm not going to ask you about the omega numbering system because we're not going to get into detail about
the 1
this.
represent or
The 1 just tells you there's 1 double bond here.
Oh, and the 9 is where is it?
Yes. Yep. Yep. So you'll definitely see something like that. So here's a list of a bunch of saturated fatty acids, right? Yeah, so 12 is very common. Lauric acid in here, but yeah, it's 12.0, 12 carbons, no double bonds. 14.0, you know, myristic acid, palmitic acid, stearic acid. So I'll talk about these, you know, like and then you have things like arachidic acid and lignoceric acid. We'll talk about arachidonic acid because that plays a role in a lot of stuff in here. And arachidonic acid has 4 double bonds in it. So that's, you know, what's the difference between arachidic acid, arachidonic acid, arachidonic acid, and so on. So you can get crazy. That's why I like the numbering system in there. Okay. But I won't ask you to memorize all these common names or all the systematic names. Okay. So omega fatty acids are essential nutrients. We need them, but we can't synthesize. So things like DHA, EPA in there. So we get these in there, are omega fatty acids from fish oil capsules, or I don't know if you ever had a grandmother. It's like, oh, you're sick, have a spoonful of cod liver oil. Yeah, because it tastes so delicious. I'm not sick anymore. All right. When we look at fatty acids, you know, shorter fatty acids with that carboxylic acid group can be soluble in water. But the longer that chain is, the longer the tail region is, the less soluble they become. Okay? Our melting point will decrease as the chain length decreases too. So the shorter the chain, the easier it is to break them apart. We also see the more we increase double bonds, the melting point will decrease as well too. Because most of our double bonds, in fact, all of our double bonds occurring naturally are cis, so they introduce this kind of kink into the chain. And if there's a kink in there, they won't pack together as neatly. Okay. So our saturated, we can do this nice little shorthand drawing here. In fact, I used to like give you the nomenclature, like 18:3 delta 9, 12, and 15. Draw that. You know, maybe I'll give you this and give me the shorthand. You know, I'm not going to say, tell me this is palmitic acid or whatever this happens to be.
This just looks like a skeletal structure.
Yes, yeah. So it's another reason why I like these. They're very easy to draw. I mean, basically everything on here is hydrogens. So we're just putting this little skeletal backbone in here with a carboxylic head group. Okay. Whenever we have a double bond that occurs naturally, it will be in the cis configuration. So when it's in the cis configuration, you get that kink in there, right? If it were in the trans configuration, it would look extended like this conformation, and we'll see that coming up a bit. Okay. So saturated fatty acids tend to pack very orderly. You've got that hydrophobic effect. They're going to kind of stick together there. All those hydrophilic head groups, those carboxylic acid head groups, will line up together, and then these can sit in here and kind of, you know, be protected from water as they line up together. For saturated fatty acids, they're very orderly because they fit together. They stack pretty nicely. On the other hand, our unsaturated fatty acids with these cis double bonds in here, they can't line up as well because that kink prevents them from fitting together nice and, you know, evenly. So they tend to have lower melting points. So the more double bonds that we have, the lower the melting point. Okay. You can see where there's going to be a question about this. Trans fatty acids are formed by partial dehydrogenation of unsaturated fatty acids. Okay, so we can actually do this. So to make like cooking oil that's shelf stable, trans fatty acids really last a long time. They won't go rancid. Have you ever had like really old butter that's been sitting out for a while and you can smell it's going bad, it's oxidized because of the accessibility in there. But a trans tends to last a lot longer. The trans double bond allows it to have that extended conformation. So you can make them as oils. They tend to be more liquidy in there too. They have higher melting points than the cis forms. But one thing that was discovered in the 90s is that if you consume a lot of trans fatty acids, it increases the risk for cardiovascular disease. We won't get into it this semester, but when it comes to metabolism of fatty acids, our saturated fatty acids are actually monounsaturated are pretty good. Even there's a nice enzyme that will cleave off those carbons we use for energy fairly easily. Cis bonds are great for that. The trans bonds are a little bit harder to deal with. So what they would tend to get is inflammation and plaque formation and lead to an increase in heart disease. So I like this. Current trend, reduce trans fatty. Okay, all major fast food restaurants have ceased using trans fatty acids for at least a decade now. So, maybe we need an 8th edition. No, okay, just kidding. No. All right, so yeah, fatty acids, glycerin. So I'm not really going to ask about this categorization, but this is a good thing to kind of look at. But we're mainly going to talk about fatty acids, glycerolipids, glycerophospholipids, and sphingolipids, and sterols. And the rest we won't talk about too much. All right. So the last little thing I want to talk about today is our triacylglycerols. So free fatty acids are one thing, but in an organism, we don't see free fatty acids. We only get free fatty acids when they are being metabolized and you're cleaving off those carbon groups to produce energy, or when you're building them up to store them later. So most fatty acids in biology are going to be found as our tags, our triacylglycerols. Solid ones we call fats, liquid ones we call oils. This is also primarily the way we store them in our adipocytes in here. They have very low solubility because we have this esterification of the carbonyl head group. So we'll see that come up in a picture here on the next slide. So very, very low solubility and very low density. They tend to float. And I don't think we talk about this too much. That's next semester. Our cholesterol metabolism. When you consume, say I go to Wendy's and eat a big old bacon cheeseburger or something like that, all those lipids that come in to my body will get into the gut as chylomicrons, which have are huge and very low density, and they float. They then we'll see them go into VLDLs, very low density lipoproteins, and get transferred to LDL, which is the bad cholesterol. It's not really bad, but that's what we've seen a lot of plaque formation. There's an intermediate density lipoprotein, but that's a transient form. And then there's HDL, or high density lipoprotein, which is where you tend to scavenge our cholesterols and bring them back to, so LDLs kind of take them out, HDLs bring them back. And it just really has to do with the density. So all of our fatty acids and triacylglycerols tend to be less dense than proteins. So they will float on top.
So it's good to have some body fat on us.
Yes, yeah.
That means we're storing lipids. So somebody that doesn't have any body fat on them, are they unhealthy?
Yes, there is a mutation that causes people to be unable to either produce or store triacylglycerols. And I mean, you can live, but they're very sensitive to cold. There's some other health issues in there. They look just absolutely emaciated. But you can't store energy for long periods of time. So, and in fact, I need to look at that because I can't remember if it's this semester or next semester. But I have a question about that. So we may try and do a case study. I'll have to look at my time frame and how many weeks we have. But I think we are a lecture or two ahead, so I may try and pull that out. But there's a condition where people can't store body fat. But as far as health goes, if you have high LDL readings or high triacylglycerol circulating, that's generally associated with a poor health outcome. I always like to pull in my data, like my LDL is just in the bad range. My HDL, well, I need to work on it, but when I originally started lecturing this, it was like 60% above normal. So really the ratio of HDL to LDL is an indication of healthy cholesterol metabolism. So you can have high LDL as long as you also have really high HDL. That just basically means you have a lot of stuff circulating and going through the system there too. If you have high HDL and low HDL, you have a lot of cholesterol going out, but not a lot of it being scavenged and taken care of. So it's more likely to lead to plaque formation, macrophages in there, and arterial blockages, cardiovascular disease. So one solution is just have low of everything. Another solution is, as long as that ratio is healthy, you're good. My ratio has been declining, so
they haven't found anything to help with people who can't store body fat on.
Right, I think. So, like I said, it's a genetic mutation. I'll have to go back and look at it. I'll have to see if I can find it. That's what I'll look for right after class. I'll have to see if I can find that. But it's a genetic mutation where they don't have the framework to do that. So
basically, they need to do research more.
Yeah. You would need to have like a gene therapy maybe to correct whatever error is being made.
Okay, so you don't think it's like a medicine they can take?
No. Yeah. So usually I think it's just they treat with diet, high sugar diet. I always think of like people I've met with different medical conditions. There's one guy who couldn't maintain his salt balance. So the doctor's like, eat lots of bacon, eat all the bacon you can because you get lots of salt and lots of fat in that way. So that's unusual, rare conditions. But I'll look for that. And maybe we can talk about that on Tuesday. All right, so our triglycerols typically look like this. All right, usually, so we're going to have this glycerol backbone. Good to know the structure of glycerol. So, oh my, one more structure there. Only 4 structures. Not too bad, right? 4 or 5 structures. Yeah. Okay. So our glycerol is our backbone in there. We've got these 3 hydroxyl groups that can be active sites, and we're basically going to attach 3 fatty acids to those. So we're going to see this condensation. We get rid of those protonatable hydrogens. We now have this ester linkage in here, so it's much less soluble. Typically what we'll see is the outer 2 fatty acids will be saturated, and the inner one will be unsaturated. Sometimes polyunsaturated, sometimes mono. Okay. The advantage for this of fuel storage in here, fatty acids have more energy per carbon because they're more reduced. Okay, so we got more energy there. There's less water because they're nonpolar, so they don't need to be dissolved. Glucose and glycogen tend to be short-term to medium-term energy needs. They can do that really quick delivery. Fats will be long-term. This is why when they talk about like how long can you survive in the wilderness, you can go 2 or 3 days without water because you got to have water to be flow through and all that stuff in there. But you can last weeks to months without food because you can just break down the fat over time. If you think about various diets that are out there, a lot of people like the keto diet or the Atkins diet or whatever variation that they've renamed it in here too, where you essentially have no carbohydrates. So if you cut out all carbohydrates, the idea is it will stimulate your body to burn the stored fatty acids in there. It is sound, but if you cut out all carbohydrates, one of the things that happens is you start producing acetone as a side effect in there, so that you can have ketoacidosis that will happen. So especially if you're like diabetic, I've got a great, dang it. We've got a great question for Biochem too. Talk about you've got a friend who has diabetes who decides to start the all bacon diet. What health risks would you be concerned about for your friend? So fun stuff like that. You can get into that, but we're not there. We're just going to talk about this here. So fats are a higher source of energy that can be stored over a longer period of time. Slow delivery as opposed to fast delivery like our glucose. It's like your little power bars will have lots of sugar and protein in them. Longer term, you get fat in here. This is an example of adipocytes. So we can see basically we have these big giant fat droplets in here. It's kind of like, I always think of little cells as like ticks that have swallowed blood because they're just all swole up with fat in there. All right. Waxes, just definitionally, waxes are esters of long chain saturated fatty acids. So we basically take a couple of these fatty acids and kind of hook them together at the head group, and we get a wax out that way. So our beeswax in there. they tend to have very high melting points, so they're going to be solids at room temperature, whereas our triisoglycerols would be liquids or sometimes solids in there too. But our waxes can function as waterproofing for feathers, pliability in the hair, prevent evaporation. You see like waxy leaves will prevent the water from evaporating from those. For example, if you like grow gardens, like if I didn't water my peppers and my tomatoes, you can see their leaves kind of wilt and droop like that. If you look at tropical plants, they're often waxier, so even when there's not water, they still retain their shape and they don't lose water that way. And of course, you can see our wax is used in all sorts of cosmetics as well too. There's our beeswax. We got a bunch of different lipids in there, but I'm not going to ask too much about waxes. Maybe what is a wax? What's a wax? All right. I don't want to go here. Nope, I don't want to go here yet. So one thing to keep in mind is this structure is very, very good. Where we're going to go on Tuesday is we will talk about glycerophospholipids and our structural lipids and membranes. And we're going to see some very similar structures in here. We're just going to change, we're going to remove one fatty acid and put up a phosphate usually there. But I think this is a good stopping point. Yeah, I got like 6 minutes left anyway, so give you guys a little extra time. I will go look for my case study on people who cannot store fat. There's all sorts of really neat case studies out there. That's why I like biochem too, so you can get into that type of stuff as well. And hopefully by the end of this weekend, I'll have all the exams graded and provide you guys all sorts of feedback. Yeah, I know. Hopefully this will be more fun. We're getting into, definitely exam 2 is the hard exam. All right. I guess that is it.